Hi, my name is Brittany Forte, and I'm a second-year cancer biology PhD student in the campus lab. The campus lab studies the most commonly transmitted sexual infection, HPV. Human papillomavirus infections are usually cleared by the immune system, but when not, they can become persistent infections and lead to deadly head, neck, and genital cancers in both men and women. Thus, we have the goal of better understanding how HPV might evade and antagonize the immune system in order to avoid clearance. My project centers around the hypothesis that HPV's unique intracellular trafficking mechanism might enable the virus to evade detection during initial infection. Specifically, I'm looking at the evasion of cytosolic DNA sensors, a facet of the innate immune system geared towards recognizing foreign genetic material, such as the DNA of HPV. Although active when cells are not dividing, human cytosolic DNA sensors are predicted to be off during mitosis, when cells are dividing and our host DNA would no longer be protected within the nucleus. This would make sense because you wouldn't want an immune reaction every time your cells divide. Further, a cellular organelle called the Golgi completely changes morphology during mitosis, in that it vesiculates or completely breaks into tiny pieces. The Golgi is an important landing dock upon which cytosolic DNA sensors assemble when they're activated, and therefore we hypothesize that the Golgi, an intact Golgi, is important for CDS, cytosolic DNA sensor activation, and that a vesiculated Golgi, such as that during cell division, is not sufficient to support activation. Further, work from our lab has showed that HPV strategically protects its genetic material until mitosis, when it may be safe to come out of hiding because we believe these cytosolic DNA sensors are off at this time. To test our hypothesis, I'm looking at cytosolic DNA sensors in human keratinocytes. And first I've shown that they're both present and able to be activated in human keratinocytes upon transfection with foreign DNAs. When I transfect with foreign DNAs, we see a dramatic increase in phospho-IRF3 expression. That's a marker of cytosolic DNA activation. Next, I showed bioimmunofluorescence, which is a technique used to visualize proteins within cells, that two members of the sensing pathway change location within the cell upon transfection of foreign DNA. Therefore, I can use these human keratinocytes to both see and activate these cytosolic DNA sensing pathways. Next, I wanted to look at the Golgi and how its morphology changes the activation of these sensors. I used different drugs to change the morphology of the Golgi. When the Golgi is intact, such as when cells are not dividing, foreign DNA transfection is able to turn on phospho-IRF3. But when the Golgi is completely vesiculated or in tiny pieces, such as during cell division, and we transfect foreign DNA, we do not see activation of phospho-IRF3, indicating that indeed these pathways may be off during cell division, and this may allow HPV to avoid detection. Overall, our results suggest an early evasion tactic utilized by HPV to achieve infection, persistence, and eventually cancer.